So this is about the topic epistasis. So some topics are very difficult to understand, but when you use some innovative ideas and when you use the charts and models and put your efforts in making these in the form of some kind of models or charts, you can understand this topic in a better way. So this is again a new chart which is made by the student. So the topic is epistasis, that is, it is due to the interaction of genes, or you can say that in Mendel's law there was a law of dominance, and you can see the Mendel's laws and their ratio were normally different. But in case of non-Mendelian inheritance, you can see there are modifications from the Mendel's ratio which he found at that time. So the modifications of Mendel's law ratio is the non-Mendelian forms. So in epistasis, you can see that there is interaction of genes, and due to these interaction of genes, this can be of two kinds. First is dominant epistasis and second is recessive epistasis. So here the two examples are given. First example is of cucurbits and second example is of this is the mice. So after the Mendel's, the William Batson, this one is the William Batson, who gave the term epistasis. So the epistasis means how one gene can hide the effect of another gene at other locus. So this is how the gene hiding the effect of some other gene at some other locus. So this ratio is different from the simple Mendelian ratio. So this one the Batson who coined the term epistasis. So you can explain with the example of these examples of cucurbits and mice. So it means gene that mask the other gene, that is epistatic, and gene that gets masked is known as the hypostatic. So a gene at one locus hides the effect of another gene at another locus. So it is of two kinds, dominant epistasis and recessive epistasis. In case of dominant epistasis, the dominant allele hides the effect of another gene. So you can see fruit color in summer squash cucurbita. So where capital W is showing the white color, small y or small w is green, and capital Y is always yellow. So there are three kinds of genes: capital W, capital Y, small w and small y. So in recessive epistasis, this recessive allele that hides the effect of another gene. So this is a coat color in mice. Capital B is black, capital small b and capital C is brown, and small c is the albino. So three types of varieties are formed here. So you can see here how beautifully she has tabulated in a single frame, that showing the interaction of genes in the dominant epistatic and in the recessive epistasis. So you can see, you can just elaborate this with the example that in dominant character, this one, dominant epistasis, gametes are formed like capital W, capital Y, capital W, small y, small w, capital Y, small w and small y. So when there is even a single capital W, then it will be the white, and when there is single capital Y, then it will be yellow, and when there are both recessive W and Y, then it is a green. So ratio is changed here. So you can see the ratio that is given here in this one dominant, that is the white, yellow and green, and ratio will be different here. So you can see here 12:3:1. So 12:3:1 in case of dominant epistasis, you can see here. So one while one masking the effect of other gene at some other locus. So it is turning it into single Y is yellow and capital Y is yellow, and when there are double recessive, then it is the green. And in case of recessive epistasis, you can see the example of mice, coat color in mice. When capital B, then it's black. When small b, then it's brown. And when C, small c, then it is albino. So you can see here the ratio will be 9:3:4. 9 will be black, 3 will be brown, and 4 will be albino. Albino means where the small c is double recessive character, and black is when double capital B is present, and brown is when small b is present. You can see small b here. You can just check these. So these are the examples of recessive epistasis. So you can give both of these examples in case of epistasis. So this is beautifully designed chart giving the interaction of genes in the form of dominant epistasis and in the form of recessive epistasis.